Hey there. Today I'm diving into Gel Permeation Chromatography, or GPC for short. GPC is a form of size exclusion chromatography primarily used to separate molecules based on their size in solution. The technique relies on a porous stationary phase, typically consisting of polymer-based beads. When a solution containing analytes of varying molecular sizes is passed through the column, smaller molecules penetrate the pores of the stationary phase, experiencing a longer path and eluting later. In contrast, larger molecules which are excluded from entering the pores elute first. This separation allows the determination of molecular weight distribution, a critical parameter for macromolecules. The elution profile is usually correlated with molecular size using a calibration curve generated from standards of known molecular weights. GPC was first introduced in 1964 by Moore of the Dow Chemical Company, marking a pivotal advancement in the characterization of polymers. Its development was driven by the need for an analytical technique capable of resolving the complex molecular weight distributions of synthetic polymers. If you're into polymers, biopolymers, or proteins, GPC is your go-to technique. So, what's the principle behind GPC? GPC operates on the principle of size exclusion, where molecules are separated according to their hydrodynamic volume in solution rather than their chemical properties. The hydrodynamic volume, influenced by molecular weight, shape, and solvation, determines how a molecule interacts with the porous stationary phase. Larger molecules are excluded from entering the pores of the stationary phase and travel more quickly through the column, eluting earlier. In contrast, smaller molecules penetrate the pores to varying extents, resulting in a longer retention time due to their more tortuous path through the column. The elution time in GPC is inversely proportional to the hydrodynamic volume of the analytes. Molecules are eluted in descending order of size, with larger molecules appearing in the earlier fractions and smaller ones in the later fractions. This relationship is typically represented as a calibration curve, where the elution volume or time is plotted against the logarithm of the molecular weight for standard compounds. This enables the determination of molecular weight distribution for unknown samples, assuming a similar correlation between molecular size and weight. The stationary phase in GPC columns consists of inert cross-linked porous gels made from materials such as polystyrene divinyl benzene, silica, or polymethacrylates. These gels are engineered with specific pore size distributions to target the separation range of particular molecular weights. The mobile phase, a solvent compatible with the analyte and stationary phase, plays a critical role in facilitating analyte movement and maintaining molecular solubility. Common mobile phases include organic solvents for synthetic polymers and aqueous buffers for biopolymers. The interplay between the mobile phase dynamics and the stationary phase ensures reproducible and efficient separation, making GPC an indispensable tool for analyzing macromolecular samples. Now let's talk components. The chromatographic column is the heart of the GPC system, where the porous material used for packing determines the separation efficiency. The choice of packing material is based on the molecular weight range of the analyte and chemical compatibility. Commonly used packing materials include polystyrene-based gels, silica-based gels, and polymethacrylate gels. The mobile phase serves to dissolve the analyte and transport it through the column. The choice of solvent depends on the solubility of the polymer and compatibility with the stationary phase. Selectable solvents include organic solvents and aqueous buffers. Detection is critical for analyzing the molecular size distribution of eluted fractions. Key detection systems include refractive index detectors, UV detectors, multi-angle light scattering, and differential viscometers. And don't forget the pump, the heart of the system, pushing the mobile phase and sample through the column. Pumps ensure stable and precise flow rates, typically in the 0.5, 1.0 mL/min range, which is critical to maintaining consistent elution times and separation efficiencies. Syringes introduce samples into the column in a reproducible manner, minimizing sample spreading. An automated injection system improves reproducibility and reduces human error. Together, these components enable efficient, reproducible separations of large molecules, making GPC an indispensable analytical technique. Next, we understand the GPC experimental workflow. First is sample preparation. Proper sample preparation is essential to ensure accurate and reproducible GPC results. The sample is dissolved in a solvent compatible with the analyte and stationary phase, usually the same solvent used for the mobile phase. Dissolution ensures that macromolecules are in a solvated state and prevents aggregation or incomplete separation. The dissolved sample is then filtered using a membrane filter to remove particles that could clog the column or interfere with the separation process. This step also minimizes baseline noise in the detector. Next, a calibration curve is established using standard polymers with well-characterized molecular weights. These standards cover the expected molecular weight range of the sample and are injected into the GPC system under the same experimental conditions. The prepared sample is introduced into the GPC system via a feeder, typically using a fixed loop to ensure reproducibility. The injection volume is optimized to balance the detection sensitivity and avoid overloading the column, which may compromise separation efficiency. During the run, the detector continuously monitors the eluted components. The data acquisition system records the detector response as a function of retention time or elution volume. A chromatogram is generated showing peaks corresponding to the separated components of the sample. The system's workflow ensures accurate characterization of macromolecular samples in GPC. What about the pros? GPC provides detailed information about molecular weight averages and the polydispersity index, essential for understanding polymer properties. The process preserves sample integrity, allowing for downstream analyses or the use of separated fractions. GPC is applicable to a wide range of macromolecules, including synthetic polymers, biopolymers, and proteins, with customization possible through solvent and stationary phase selection. Automated systems and standardized protocols ensure consistent results, vital for quality control applications. GPC is also suitable for both analytical and preparative purposes, enabling detailed characterization or the isolation of specific molecular fractions. Integration with detectors like MALS, differential viscometers, and RI expands the analytical scope, providing insights into molecular conformation, branching, and intrinsic viscosity. Finally, applications. In polymer science, GPC is the tool of choice for analyzing polymer materials. It can determine the average molecular weight of a molecule as well as the polydispersity index, helping us to gain insight into the relationship between the structure and properties of polymers. In addition, GPC can effectively monitor the progress of polymerization reactions, providing important data for optimizing synthesis conditions. The technology also allows for the separation and characterization of copolymers and polymer blends, providing the basis for the development of new polymer materials. GPC also has a wide range of applications in the biochemical and pharmaceutical industries. It can separate and purify biomolecules such as proteins, peptides, and polysaccharides, ensuring high purity and functional stability of drug molecules. In the biopharmaceutical field, GPC is used to detect the formation of protein aggregates to avoid immune reactions caused by aggregates. In addition, in the study of drug delivery systems, GPC is used to analyze the molecular weight distribution and degradation behavior of polymer carriers to ensure controlled drug release rates and effects in the body. Material science is another important application area for GPC. It is used to evaluate the molecular weight and distribution of elastomers, resins, and nanomaterials, providing data to support the optimization of material properties. For example, when developing high-performance adhesives or conductive polymers, GPC's accurate analysis can help us develop more reliable materials for industry. Finally, the field of environmental research also benefits from the use of GPC. In microplastics research, GPC can identify the polymer types of plastics and their degradation products, providing a scientific basis for combating plastic pollution. GPC can also be used to analyze molecular changes in degradable polymers to assess their degradation properties in the environment. Overall, GPC is a cross-field core analytical technology, and its high efficiency and accuracy make it play a crucial role in polymer materials, biochemistry, material science, and even environmental protection. With the continuous progress of science and technology, I believe that GPC will show its value in more fields and inject new impetus into our research and production. Whether you're a seasoned researcher or just getting started, understanding GPC can elevate your work. Thanks for watching.